Thank you for joining us for today's seminar. In case you don't know me, I'm Jeff Bradshaw. I'm the new director of the Dr. Plant Health Program. Very glad to be introducing Allie Loker here this morning. She got her BS degree in community environmental sociology at University of Wisconsin-Madison in 2012. Following her graduation, Allie worked for Jen Air Family Farm, Stone Creek Farmstead, Blue Moon Community Farm, AmeriCorps in Lincoln, and then finally worked for the IPM Institute of North America before making her decision to further education and experience with the DPH program here at UNL. Allie enrolled in that program in May of 2019. And during Allie's tenure in the DPH program, she held internships with Nestle, Gerber, as well as Arbor Day. And she's going to talk about those experiences here this morning as a part of her seminar. Allie, take it away.
Okay. Oh, thank you. Okay. I'm just paranoid about this Zoom audio now. Okay, I think it's working. Thank you all so much for coming. I feel quite overwhelmed by your presence and support throughout the process, and including today. So really do appreciate you coming. I'm excited to share the work of my doctoral document entitled Assessing Functional Biodiversity for the Future of Plants, Planet, and People. But before we start, I really must thank the folks on the screen, as well as many others who've really helped me throughout my life, and especially in this program. First and foremost, my family who's sitting here in the front row, my parents, Rex and Julie, and sister Emily. They've traveled from out of state and they're in the front row, which is very metaphorical for the ways that you've shown up for me in my life. So thank you. My dog Mochi, who has provided essential emotional support. He could not be here because I scheduled this during his nap time. So, but he's here in spirit. Thank you to my friends and DPH classmates. Some friends can really empathize with the graduate student experience, and others have reminded me that there's life outside of grad school. So both very important, particularly DPH classmates. You're the only ones that know what this is like, and it's a trip, and I'm excited to get out there and make an impact with you all. I had the pleasure of working with not one, but two DPH program directors in my time. Dr. Gary Hine got me through the first 3.5 years of the program. Thank you for encouraging me to make the program my own and supporting me in doing that. And then in my final semester, Gary retired, congrats. And Jeff stepped in and said, hey, yeah, I'll take you over the finish line. And so, too much fun writing this document with Jeff, and really appreciate your support through this final leg. And our admin associate, Kelly White, is a joy of a human and a gift to our department. She supports students above and beyond, and I always leave her office in a better mood than when I came in. So thanks, Kelly. My committee members, Dr. Sam Wortman, Chuck Francis, Lauren Giesler, and Jerry Adams. Thank you for your mentorship and support and connections to research and teaching opportunities, internships, the whole gamut. I'd also like to thank Sue Wortman, who connected me with my internship at Arbor Day Foundation, and Barb Francis for always welcoming me into your garden, your kitchen table, and for letting me wear my Wisconsin sweater to Husker volleyball games. And I would like to next thank my colleagues at Gerber and the Arbor Day Foundation from my two internships that I did through the DPH program. Those have been really valuable experiences to concretely apply and synthesize the knowledge that I've learned. To UNL faculty and staff, thank you for offering instructive courses and different perspectives that I've really carried through with me. And all of you, I already thanked you, but again, thank you for being here today. And so at the Arbor Day Foundation, we always like to start with our why. What brings us to this work? And I thought that might be a nice way to start introducing who I am and why I care about this particular topic. So I've always been interested in being outside. You haven't seen this picture yet. This is one of my first plant walks. I'm sure saying something profound to my sister, my mom, and our family friends. But in all honesty, I've been fascinated by the natural world and all that it contains. that fascination with nature led me to work on several farms throughout my undergrad and post-undergrad experience. And I really found that a fascinating way to connect people and science and food, which were great loves of mine. And who doesn't love harvesting sweet potatoes the size of your face? And through the DPH program, I've really been able to gain a deeper knowledge and breadth of knowledge on the topics that are really important to me. And this is a photo from my internship at Gerber, where I got to run around collecting insect samples and soil samples for biodiversity assessments. And I'll speak more about that later. But that's really where things started connecting and clicking for me. And lastly, being able to share, I'm so excited about science, being able to share the results of science and translate them to different audiences is really something that I'm also passionate about. So really, in summary, my why is connecting people with science to create a more sustainable world. And so over the last 3-ish months, I've been really diving into the topic of assessing functional biodiversity. And this is an outline both of my paper and my presentation today. So I always like to start with what is this and why is it important? I'll talk a little bit about more of the tactical how of how functional biodiversity is assessed in agroecosystems and in forests. And then I have a sociology undergrad, and so I'm always curious about how people play into the equation. And so I'll conclude with a discussion of how participation in biodiversity assessments can help address some barriers and opportunities. So to kick us off, a little bit of this section will be making sure we all have a common understanding to build from. So some of this y'all might already know, and it might be new to others of you. So biodiversity has many different definitions, but I really like this one from the Convention on Biological Diversity. And they say biodiversity is the variability among living organisms from all sources. And we can think of it as diversity within species, so genetic diversity, diversity among and between species, species diversity, and then at the ecosystem level as well. And I think one of the things that really resonates with me about why biodiversity is important is that it really highlights the interconnectedness that all species have on this planet. And I love this quote from Chief Seattle, who says, quote, "Humankind has not woven the web of life. We are but one thread within it. Whatever we do to the web, we do to ourselves. All things are bound together, all things connect." And this is juxtaposed on this slide kind of awkwardly with this Jenga game. And this is an educational tool that I think really highlights the interconnectedness of species and biodiversity. So educators have adapted this game where each block in the tower represents a species. And so if you're familiar with the game, players take turns removing each of the blocks. The tower becomes increasingly unstable until it collapses. And so in this context, each of those removal of the block would represent an extinction of species. So a couple of species go extinct, maybe the tower can still stand, but at the end of the day, it will eventually collapse. And that segues into what is happening on our planet. We are seeing global declines in nature, and humans have really played a role in that decline. And so these are statistics, pretty sobering statistics from a few global assessments on the state of biodiversity. So we can see that about a quarter of the species, which represents about 1 million species, are threatened with extinction. Significant declines in natural ecosystems and the abundance of terrestrial species. And then indicators that have been developed by Indigenous peoples and local communities show ongoing deterioration. So this is a lot. So I do want to say that things are bad. And also we are trying to set goals to conserve biodiversity and mitigate this crisis. However, our actions have so far not been enough. We've set global goals, the latest of which were the Aichi biodiversity targets, which was a 10-year plan to protect and conserve natural systems. And you can see based on some of these headlines, we did not meet those targets on a global level. There's a little bit of progress at a national level, but overall, we still saw that the indicators were getting worse. And so in December of 2022, nearly 200 countries signed a new global biodiversity framework. And I will note that the US was not one of them. And for me, this question outside of why wasn't the US one of them is how will we know if we're making progress? How will we know if we're doing better to meet those goals than we did in the past? And that's one area where I think biodiversity assessments can come into play. So assessing biodiversity can help us prioritize our efforts. We have limited resources. And so I think that the results of biodiversity assessments can help us target those resources where they are most effective. And then ultimately, again, assess how effective our interventions are. And so these images highlight some of the different stakeholder groups that are involved in this process. So local groups, governments, non-governmental organizations, and the private sector can all be involved in various combinations and to various degrees. And something that I find really fascinating is how do we integrate the different values and goals and priorities of these stakeholder groups? And so I'm going to stop with that discussion there and pick it up at the end of the presentation, but I wanted to frame that a little bit for you all now. Biodiversity is connected to a couple of other concepts that I want to make sure we have as part of our foundation. So this flow chart is a very, very basic way that people are connected to managing ecosystems. And I've put sustainably in parentheses. And so our management then can have a positive or negative effect on biodiversity, which in turn affects ecosystem services, which are benefits that humans receive from the ecosystem. And so this relationship at a high level is well understood and accepted, but the details of kind of how and exactly what happens among these relationships is a feature of an opportunity for a lot of future research. And for me, that gets me really excited. Like, we don't know exactly what is in these arrows and how it all works. And I think assessing biodiversity can play a role in that as well. And so biodiversity itself can be categorized in 3 different ways: the composition, structure, and function of an ecosystem. So composition is who's here, what species are present. The structure asks how the organisms are connected to one another. And then the function is what are those organisms doing? And specifically looking at for ecosystem processes and ecosystem services. And these are all interconnected, so I'm not saying we should only assess functional biodiversity, but that it should be a component here. But today we are going to focus on functional biodiversity, and I decided to focus my paper on that because I think it can be a really compelling argument for funding. If we can say conserving biodiversity will give humans these things, so give us money to help do that. Additionally, more on like a personal level, I think it's exciting to be able to use functional biodiversity as a tool to increase our awareness of those interconnections. And then my hope would be once we see and really feel those interconnections, that we'll eventually make our decisions more in a reciprocal way with nature versus extractive. And so functional biodiversity is defined as the range and value of species and organismal traits that influence ecosystem functioning. It's a little bit of a mouthful, but really what we're looking at is how are organisms related to ecosystems in a mechanistic way and related to their function. Functional traits are features of the organisms that directly contribute to their participation in those ecological processes or services. So within those organisms, there are specific characteristics or traits that help them participate in the ecosystem service. And then we can look at functional groups, which are groups of organisms that provide a similar ecosystem service within a community. So all these organisms each have functional traits, and then that group of them contribute to the ecosystem service. And this can be done, this assessment process can be done in 6 steps, which is a result of my literature review and kind of combining different guidances. But I think that this process is general enough to give us a start here. So first, we want to establish the goal or purpose. And next, identify those ecosystem services, so the things that humans are benefiting from, which ones are important to assess. And then sorting our functions into groups and categories, looking at those traits, which are what we will then directly measure, weighting the traits according to their relative functional importance, And then lastly, taking those raw measurements and aggregating them into indices, because that can help us explain and predict ecosystem processes, and also makes it a lot easier to communicate to other stakeholders. And so I'll be walking through this step by step, so don't worry if some of it's a little fuzzy right now. But to conclude this section, I hope that we're all on the same page about the importance of biodiversity to ecosystem services, which are critical for our lives as humans and also the other lives on the earth. And these interconnections and relationships are complex, and there's a lot of exciting work, I think, to still be done there. And functional biodiversity specifically can help us explain and predict ecosystem processes and ultimately inform our decision-making and conservation efforts. And so this portion of the presentation is a little bit more tactical, so I'll walk us through that 6-step process and give some examples from how this is done in agroecosystems and forests. And I'll also share experiences that I had in each of my internships through the DPH program, which are conveniently in agroecosystems and forests. So just a quick bit of background looking at biodiversity specifically in agroecosystems. So agroecosystems are ecosystems that we manage as humans for agricultural production. We can divide biodiversity in these ecosystems into their components. That's the who's there, and then the functions, things like nutrient cycling, water quality, things like that. And then on the right hand of the screen, you can see that we have agroecosystem services that are related to biodiversity, and we have management that affects all of that. And so really the key here is that biodiversity and the ecosystem services in agriculture are related to one another, and management influences both of those components. Similarly, forests are home to a lot of biodiversity, and there is a spectrum of management that can influence biodiversity in forests as well. This figure here shows a number of icons that are emblematic of ecosystem services we receive from forests. So timber production, recreation, habitat for animals, on and on. And again, we have different levels of management from very minimally managed, more natural forests to heavily managed timberland. So biodiversity will look different in each of those cases. I'm going to next walk us through the 6-step process with examples from real functional biodiversity assessments in these two contexts. So the first 2 steps I've lumped together because I think they're quite interconnected. A broad overarching goal of functional biodiversity assessments is to understand the effect of diversity within the functional groups on the ecosystem services. And so when we're thinking about the goals, this is like kind of the big goal, and then each assessment might have a more specific goal. And then identifying ecosystem services to be assessed. So these examples are from functional biodiversity assessments of soil, plants, and arthropods. And I've just thrown up some example goals. So we might be looking at environmental sustainability of farms, or broadly ecological processes, or the effects of a management practice like forest buffers. In terms of example ecosystem functions or services, in soil, we might look at soil enzymatic activity or nutrient cycling. For plants, we might again look at nutrient cycling, but how the plant is responding. And then in arthropods, we might look at things like biological control for our ecosystem services. And the goals that we set out influence what ecosystem services then we will affect, and it's kind of a cascading effect throughout the assessment process. So this is a really key part of the assessment stages. When we're looking at functional traits, I will couch this all with saying functional traits are not a big component of biodiversity assessments generally. So this is an area where we're finding new ecological traits or functional traits all the time. And they're just generally underrepresented right now. But broad guidance here is that they should be quantifiable, easy and cost effective to assess, repeatable and ecologically meaningful. I really like that last category, the ecologically meaningful one. It's very important and easy to overlook though. So examples for functional biodiversity assessments in soil for functional traits might include things like enzymatic activity, entomophily, Plants, we might look at things like specific leaf area or plant height. And then arthropods, we might look at things like dispersal method or body size. And again, all of these traits relate back to those overarching goals and ecosystem services we identified in the previous step. Measurement. This is a really cool area of a lot of innovation and development in terms of how we actually measure the trait diversity. This is also the most time-consuming step of the process. And so there's a lot of effort underway to make this a more efficient component. There is a little bit of direct observation of individual organisms that still happens as part of biodiversity assessments, but really the field is moving towards rapid biodiversity assessments and other technologies that can make it a quicker process. So rapid biodiversity assessments, like their name implies, are ways that we can make this process quicker, cheaper, more effective, and over larger scales. We can do this in a non-DNA based approach where we're looking at groups of surrogate taxa or more of a taxonomy or sampling approach. And then we can use DNA based assessment techniques like metabarcoding in combination with bioinformatics to analyze DNA of bulk samples. And when we combine that with ecological network analysis, we can start to understand the functional biodiversity component. Remote sensing is another opportunity to scale biodiversity assessments. We can use satellite imaging in combination with on-the-ground observations to really amplify the scale of our assessments. And that's particularly useful for plants because that's easier to see from space. The next step is assigning the trait weights according to their relative functional importance. And I think this is another area where there's a lot of opportunity to develop and systematize. Historically and now, this step has been done in consultation with experts, and that is valid and great. And I am not trying to diminish that importance of the human component, but it does leave room for selectivity and bias just based on our background and experiences and the lens that we see the world through. And so there's a bit of work. I found one paper where they systematically scored and weighted factors in an agroforestry system in Brazil to try to then get a mathematical indicator of system performance. And so I think that techniques like the one used in this Calocancha paper can really be explored further and applied in different situations to see if this is a way to reduce some of the bias of this step. And maybe there's a hybrid approach of involving the experts and doing a systematic scoring. We'll see. And lastly, calculating our indices. So as a broad guidance here, we want to include multiple traits, account for abundances, and measure for all aspects of functional biodiversity. Some example of indices here. Fusaro et al. Used several soil biological quality indices related to the presence of arthropods or earthworms. And then you'll see that my plant and arthropod examples both used this set of functional biodiversity indices that are richness, evenness, and divergence. And I think what's cool about thinking about ways to systematize this process is that we can compare results much more easily. It's a little bit harder to compare a soil biological index that maybe has totally different calculations from a functional evenness index. And so I think that there's some room for coming to more of a consensus on the indices as well. All right, so summarizing this subsection, I gave you some examples of how we can apply this functional biodiversity assessment process in agroecosystems and forests. And we're doing this because of the important role that biodiversity plays. And as a brief reminder of another why, that we can explain and predict ecosystem processes by assessing functional biodiversity. Okay, we're getting to my favorite part. I mean, it's not my favorite, but this is where I found a lot of joy in applying my internship experiences to this topic. And so my first case study is from my internship with Gerber in the summer of 2021. And as part of this internship, I conducted on-farm biodiversity assessments with some of their growers. This content comes from a report that I wrote for that internship, and I do have permission to share it. All the grower data has been anonymized. And these pictures, this has nothing to do with biodiversity, but if you work for Gerber, you must drive the pea harvester at least once. So I did. And then in the center is my kind of mobile biodiversity assessment office. So some sweep net, beet sheet, soil sampling, and then on the far right is some soil sampling. And so a little bit more context on this, this case study is that Gerber is a baby food company based in Fremont, Michigan. They work directly with many of their domestic growers, and they have a couple of existing sustainable agriculture programs. So one focused on integrated pest management, and then a fairly robust soil testing program as well. And Gerber is owned by Nestle, and Nestle is part of a coalition called One Planet Business for Biodiversity. And this is a collaboration that's focused on biodiversity, specifically in agriculture. And one of this coalition's aims is to enhance cultivated biodiversity. And so to meet some of these goals, because of the participation in this coalition, and then also for internal reporting, I was tasked with conducting an on-farm biodiversity assessment to establish a baseline for ongoing monitoring. And so to do this, our sustainable ag team selected 6 apple orchards and 2 vegetable farms in Western Michigan. These were all growers that supply to Gerber. And on the map, you can see that we've divided them up into regions. And I don't know how visible this is, but there are pairs of farms in each of those regions. And so one of the farms we identified as using conventional practices, and here that means that they comply with any pesticide restrictions, but don't implement any additional soil health building or advanced IPM practices. And then the other farm was identified as using more regenerative practices, so implementing those additional soil health building practices and using advanced IPM. And so that was based on the relationship that our team had with those individual growers in terms of how we categorize them. And so we looked at both above and below ground biodiversity. For the below ground biodiversity, we used a test called bee crop from a company called Biomakers. And so this company has a patented DNA sequencing and bioinformatics process that analyzes the bacteria and fungi in soil samples. And then the beneficial arthropod sampling was done every 2 weeks to collect insects that were related to pollination and biological control for our ecosystem services. And then I used 3 different methods to collect those insect samples: a sweep net, beet sheet, and sticky traps. So a bit of a discussion of the results. This is for the soil microbiome. To orient you to this table, we have our regions here on the first column, and then each row is paired with this production practice. Apples on top, squash on bottom. Unfortunately, the West Central conventional squash field results were not in at the time of the report, so they all say TBD. But the bee crop test has 3 different indices. So those indices that I was talking about, they have a functionality index, which is what are the soil microbes doing in the soil? The resistance index communicates how well that community can withstand stress due to disturbance. And then the biodiversity index is more of a traditional measure where we're just looking at who's there, how evenly or not are they distributed. And so in all cases, higher index values are desirable. Okay, so the first thing that I would like to highlight as an observation, none of this is statistical, we didn't do statistical analysis, but that across the board, we saw regenerative farms had the same or higher index ratings across all the regions as conventional farms. And so I think this is just an interesting thing to note. Remember, these are for establishing a baseline, so we're not necessarily explicitly comparing the two, but just an interesting observation there. And another thing that I thought was notable was that we had very low ratings for the resistance index, especially in the apple orchards. And so some future questions that I have related to these results focus a little bit on this outlier of the apple orchards and the functionality index. So you have a medium rating there on this farm, and my questions would be, what functional groups are present or absent in the sample for this farm compared to the other orchards? does this medium rating really matter? Will this affect their yield or the quality of their product? And if so, are there management practices that we can amend to raise that functionality index? And then in terms of this resistance rating, I'd have similar questions about the functional groups that might be present or absent in the apple orchards as compared to our squash field that did have a bit higher of a value there. I think that it would be interesting to compare, these are very different cropping systems. And so maybe there's something going on here with the amount of disturbance in a squash field, which is more frequent than an apple orchard, for example. So maybe that could explain some of those results. Looking at the beneficial arthropod sampling, I will note here that there was not a functional component because I didn't really know about this yet. But so I calculated the Shannon Diversity Index, which is a traditional biodiversity measure for the conventional and regenerative farms. So here the columns represent those two different production practices. And of note here is that there was not a consistent effect of our production practices on the arthropod biodiversity. And this could be for a number of reasons. I was able to get to all of the farms 3 times throughout the season, but that's not a very high sample size. So it would be really interesting. This is a baseline. How will this change over subsequent monitoring years? The other things could be like environmental effects. Did our best to visit at the same time of day and control for environmental variables as much as possible, but it's the real world. We can't control it all. And so again, functional biodiversity was not explicitly assessed here, but maybe in the future we could look at those 2 ecosystem services, pollination and biological control, and see how those results might compare to one another. So to summarize this case study, I have this table with our friendly 6-step process. And looking at our soil biodiversity assessment, we did incorporate functional biodiversity into that assessment, kind of by accident, but it was exciting to see that. And so really here the opportunities for future incorporation is in this goal and purpose stage. And then the traits were not weighted in any type of way with the relative functional importance. So that could be an opportunity in the future. And then you'll see in the arthropods, almost all of the boxes are checked. So there's a lot of room here to incorporate functional biodiversity if desired into future assessments. And I think something specifically here that would be exciting, since we were working with the growers to conduct these assessments, is that we could get their input on what are important functions that you think you get from biodiversity on your farm, and use that to sort of inform these first 2 steps. I think also functional biodiversity can be a tool to communicate the importance of conserving biodiversity on farms. If we could say, hey, these are the functions that are being affected by biodiversity, there might be more motivation to conserve it on farms and change production practices. All right. I feel like I'm going a little fast. The next case study is from my internship with the Arbor Day Foundation. So we're switching gears a little bit in terms of our ecosystem context, but still talking about biodiversity assessments. And so specifically, we'll look at forest carbon offset projects. I will definitely slow down to explain forest carbon offsets. But to zoom out a little bit, the Arbor Day Foundation, if you're not familiar, is a nonprofit based here in Nebraska, and we work nationally and internationally towards our mission of inspiring people to plant, nurture, and celebrate trees. And one of the ways that we do this is by transacting and investing in forest carbon offset projects. And in this field, generally, there's increasing interest in understanding the other benefits that might come from implementing a carbon project, so including biodiversity impacts. Okay, basics of forest carbon offset credits, if you are not familiar. So I will start on the left here with our trees, which is the core of this process. Project developers are the ones that actually design and implement a forest carbon project to generate carbon credits. So this can be planting new trees, protecting existing forests, or improving management practices. And the central idea is that those trees are going to sequester carbon. We can quantify the amount of CO2 sequestered, and then our standards bodies are going to verify that that happened and certify those carbon credits. On the far right are companies who would then purchase the carbon credits to offset their emissions. And then that money would go back to our project developers in this cycle. Money. I kind of chuckled. I was like, I should label that just in case. And so the first standard that I'll highlight here that includes biodiversity is called the Climate, Community, and Biodiversity Standards. This is facilitated by the Voluntary Carbon Standard, or VCS, in the corner. And this standard aims to certify projects that generate impacts to our climate, community, and biodiversity. And so this is not something that generates a biodiversity credit, but rather it would have an indicator in the carbon marketplace that this project has these extra benefits that are certified. So they could eventually have a higher price premium. And so just looking at the biodiversity section of that standard, the project developers, so the ones that are implementing those projects, are required to describe the original biodiversity conditions in the project area, what changes they might expect if the project were not implemented, and then any net positive biodiversity impacts due to the project. And so I think that there are opportunities to include functional biodiversity in this standard. It's not explicitly called out at all in the current standard, especially in this predicting anticipated changes without the project, because recall that we can use functional biodiversity assessments to explain and predict changes in ecosystem processes due to biodiversity. So that feels like a really important piece of the puzzle where we could use functional biodiversity. And then when we're calculating our net biodiversity impacts and figuring out how we're going to measure those impacts, I think it would be very important to include functional biodiversity metrics in that process. The next program is called Plan Vivo Nature, and that is run by another carbon standard body called Plan Vivo. And this is a standalone biodiversity standard and generates biodiversity credits that can then be bought and sold. The goal of this program is to direct private investment towards conserving and restoring biodiversity. Similar set of requirements here, where we're required to select a without project scenario. So what would happen in absence of this project? And then we'll choose biodiversity metrics again. And the key piece of this standard is where we're measuring biodiversity uplift or avoided loss, which is what then generates the biodiversity certificates. And so again, there are opportunities to incorporate functional biodiversity, especially when we're trying to predict what would happen without a project, which we can't really do, but we'll do our best. And I think functional diversity can help us with that. Choosing biodiversity metrics, so including functional biodiversity metrics in that process. And then in terms of measuring the biodiversity uplift or avoided loss, I think that could be a role of where that weighting process could come in. Maybe functional biodiversity metrics are a little bit more prioritized in that calculation. Those are just ideas. Lots of opportunities to incorporate functional biodiversity here. I'm not going to go through it one by one, but neither of these standards explicitly say, here's how we're going to include functional biodiversity in our standard. And that's okay. This is a really new part of the carbon offset space. And so I think now is really a great time to say, hey, here's why it might be important to consider functional biodiversity. All right, so to wrap up this kind of how tactical section, I hope that the case studies were helpful in showing that some of the potential to incorporate functional biodiversity assessments in agro-ecosystems and forests. There is a lot of development in the tools and technology space to try to use things like environmental DNA and remote sensing to help make these assessments more accessible and practical. And lastly, I think that generally these type of assessments can help us better understand those relationships in agro-ecosystems and forests, and ultimately help drive our behavior towards that more reciprocal relationship with nature. Okay, we'll talk about people a little bit now. This last section is just, I'm really very, very interested in the human component of all of our systems. I think we can have the greatest tools and technology in the world, but if no one uses them, they just sit there. So we really need to use all the tools in our toolkit here. And that's where the human component comes in for me. And so I'll present a few barriers and opportunities that I have seen through my literature search and also my experiences, and then propose participation as one way to help address some of those barriers and opportunities. So throughout this presentation, I will say, I've said it so many times now, that we're using functional biodiversity assessments to understand connections between biodiversity and ecosystem services. And ultimately, we're doing this to inform our decisions because we want to conserve and protect biodiversity. And I think when I think of decision making, I immediately think of social science and the role it can play in helping us better understand and improve those decisions. So whether those are policies or practices, towards ultimately deriving better outcomes of conservation efforts. And so my little schematic is that we have functional biodiversity assessments. And if we include social science, we can address some of the barriers and opportunities and ultimately get more robust, relevant, and effective conservation outcomes. And so some of the barriers that I've seen in assessing functional biodiversity, if you think way back to the beginning of the presentation, I highlighted those different stakeholder groups, and those are the general categories of people that are involved in assessing functional biodiversity. And each group is going to have different values and priorities, and there might be conflicts or underrepresentation of values from different groups. And so this is can be a really big challenge, especially in effectively assessing functional biodiversity. Many functional traits are still unknown, especially in groups like arthropods and other invertebrates. And we also have declining number of taxonomists entering this field. And so we have fewer and fewer people that might be the ones to identify those traits. So that's definitely a barrier to this process. And the traits that we do know, some of them are really hard to measure. They require expertise and significant resources. And again, tools and technology are being developed to try to alleviate that specific challenge. And then we have the age-old pervasive problem of trade-offs. We have limited time and resources, technology, knowledge to dedicate to this work. And so I think we tend to rely and get excited about tools and technology that will save us and solve all of our problems. And there's definitely a role for them here. And again, I think to fully optimize those, we do need to consider the human element. Additionally, we have some opportunities. So there's a ton of interest. If you Google biodiversity assessments, you will get a lot of results. And so there's a lot of groups that really want to know the answers to some of these questions. And there is funding available to do that. And I think biodiversity assessments can help us, again, prioritize that funding towards the most effective projects. Tools and technologies are being developed to help us do that more accessibly and practically. And what's really exciting in terms of opportunities for me is that the results of these assessments can help inform our management practices and ultimately conserve and protect biodiversity. And so engaging stakeholders can help us utilize some of these opportunities and eventually lead to more positive socioeconomic outcomes as well as environmental outcomes. So I think it's really important to include the people. And so participation and participatory biodiversity assessments are one way that I think we can include humans in the assessment process. And the need or demand for these types of assessments really comes from that demand for data. People want to know the answers to these questions. And also acknowledging the value and relevance of different knowledge systems. So we have science and we prioritize and value that a lot. And there's an emergence, and I think, of prioritization of different knowledge systems like Indigenous and local community knowledge. And so that's where I think participation can really make a more level playing field for different stakeholders to share information and the decisions. There are super important issues of power, governance, ownership, trustworthy of knowledge objectives, aligning, lots to go into here. I will not do that now, but happy to talk in other spaces about that. And this really emphasizes the importance of intentionally incorporating participation. It can be done at all stages of that assessment process that we've highlighted. And I will briefly discuss 3 different stakeholder groups: producers, Indigenous people, and local communities and citizens as ways that, or how they can be incorporated into this process. Okay, producers here could be agricultural producers, land managers of any kind, but their perceptions, attitudes, and characteristics will affect how likely they are to manage for functional biodiversity. And there's a little bit of social science work that's been done on this specific topic, which is awesome. One of the studies found that producers perceived functional biodiversity as complex and difficult to grasp. Maybe you're feeling that way right now with this presentation, but this is a really important thing to acknowledge in this process. There's also a range of different attitudes and ambitions in terms of how much producers want to manage for functional biodiversity. And certain characteristics can increase the likelihood that they will manage for biodiversity as well. So the take-home message from all of this is that I think it's really important to understand the human dimensions of this important stakeholder group, particularly if we're asking them to make changes to their behavior. And hopefully by doing this, we can have our results be more relevant to that stakeholder group and ultimately change behavior. Engaging Indigenous peoples and local communities. There's a great meta-analysis that has shown that this often results in more positive and effective conservation outcomes. And this particular group is a very critical stakeholder in biodiversity conservation. I think the statistic is really stark and startling that Indigenous lands represent about only 20% of the Earth's territory, but contain 80% of the remaining biodiversity. So Indigenous peoples are stewarding a lot of our remaining biodiversity. And so it's very, very important to formally engage them as much as possible, which is being done in some of our larger scale biodiversity assessments, where we're positioning knowledge from Indigenous and local communities as more equal to globally generated science. And then at a more ground level, this can be done by integrating the importance of the different organisms to local communities with our technical surveys to help us bridge some gaps. And this image on the right is from a community in Indonesia who is fighting to save the lands that they've relied on for generations and generations. They depend on the health of the river in the image as well for cooking and growing crops. And so this group is super important and their lands are under threat. So there's a lot to navigate and negotiate there. So I wanted to highlight them as well. And lastly, citizen science is an exciting opportunity to really scale this work and get citizens more engaged and excited about biodiversity assessments. And so this is a very simplified version of some work that's being done to leverage the iNaturalist database, which is a citizen science. You can input pictures of plants all across the world. So leveraging that citizen generated data with some existing databases, this is specifically looking at plant traits. And then using our modeling and predictive powers to generate global trait maps. So we may not be able to get observations from every corner of the planet, but we do have systems that exist that citizens are contributing to that we can really leverage here. So I think that's another exciting and emerging group in this field. And so I hope that you're a little bit on board with this participation idea, specifically with producers, Indigenous peoples and local communities and citizens. And this can help address a few of those barriers and opportunities that I highlighted. And ultimately, my feeling is that if we include participation in these assessments, we will eventually get more robust, relevant, and effective conservation outcomes. Okay. Here are some key takeaways that I hope that you all will walk away with today. First off, understanding functional biodiversity can really help us understand how we're connected to other species on this planet and how much we depend on one another. We're all part of that Jenga tower, like humans are just one block. So that's pretty humbling. Functional biodiversity assessments can be used to help us make better decisions and prioritize funding to conserve biodiversity. And then if we're aligning our management and policy decisions with the results of those assessments, it's my hope that we can work towards a more reciprocal relationship with humans and nature. And so I love quotes, and I would like to close with two that really resonated with me, specific to this work, and are kind of a call to action, I think. The first is from Dr. Robin Wall-Kimmerer, who is a mother scientist, decorated professor, and an enrolled member of the Citizen Potawatomi Nation. And Dr. Kimmerer says, quote, "A species and a culture that treat the natural world with respect and reciprocity will surely pass on genes to ensuing generations with a higher frequency than the people who destroy it. The stories we choose to shape our behaviors have adaptive consequences." A little bit of an ecological argument for a reciprocal relationship. And then the next quote is from Shutex Scott Martinez, who's a youth indigenous climate activist, a hip hop artist, and the leader of the Earth Guardians. And they say, quote, "It is in our nature to want to fight for the health of the planet. This life is a gift, and it is our responsibility to respect and protect that which gives us life." Thank you very much for coming and your attention. We have 10 minutes for questions or things that sparked your curiosity. Would love to hear it. And if we don't have time or you think of something later, my email is on the board.
We absolutely have time.
Yeah. Oh yeah, we do have time now. Yeah, yeah.
And the folks on Zoom. I don't know.
Some-
It's like 15 participants.
Just accolades. Yeah.
You sparked my curiosity on the question. Maybe I don't know what I'm asking you to say back.
That's okay.
The question that popped in my mind because of the work we do together is
Mhm.
what impact on ecosystem function is different based on what we do, which would be a wide scale intervention versus a more natural regeneration.
Mm. Mm.
It is rushing back into the forest having impacts on the slow development of biodiversity.
Mm-hmm. Mm-hmm. Yeah, what I hear in that, I'm not going to give you an answer, but what I'm hearing in that is like, is the tension between our desire as humans to do work quickly and that sometimes not being in balance with like the natural cycles and the evolution that happens, whether we like it or not. So I think that's a really interesting question and something that, I mean, assessing biodiversity could answer to some degree. Probably on a smaller scale, but I think that's very interesting. I'm curious too. Yes.
Curious about the term that you use, resistance, and its measurement was low in all of those situations.
Yeah.
Is that a measure of how threatened the biodiversity is in those systems? And how easily it can be, perturbed in a negative direction, or what is that exactly?
Sure. I'll repeat the question for the folks on Zoom. I'm supposed to be doing that. So the question is about the resistance index from the soil microbe analysis from the Gerber example, and what that index is communicating. So my understanding, based on conversations with the company, is that the resistance index is measuring how microbial communities respond to stress caused by disturbance. So is there like functional redundancy within that community if a stress or disturbance were to occur, that community could recover and still maintain their function? So a low or very low resistance index value, the way I understand it, is that community wouldn't be able to adapt or respond very well to stress. And so that's where I thought in the apple orchard versus squash scenario, that if you're tilling every year for an annual vegetable production, maybe those microbial communities are more selected for more resistant microbes versus an apple orchard that's less disturbed, and maybe those communities have kind of gotten comfy and might not do so well if something were to come through and disturb them. That's my understanding from that piece, but it's a proprietary index that they've developed, and so there's not a full, exactly like methods on that. But that's my understanding of the concept, at least. Yeah, you're welcome. Great question. Yeah, but
some of the work you did in Gerber dates back to people living in soil microbiome. Do you think where we're at using DNA is reliable enough for assessing functional biodiversity, saying, okay, they have this trait, we can assume that they're using that? Or do you think we need to shift the mindset more to transcriptomic proteomics to say, okay, they're actually are expressing this theory?
Yeah, I didn't find a ton in the literature speaking about the functional traits of soil microbes specifically. So I would, like, in answering your question, I would say there's a lot of room to negotiate between those more in situ observations and some of the omics techniques that we're developing. Yeah, I wouldn't put like a ton of weight in a soil sample from a single time point in a single season to make a big decision off of. So I think there'll be, maybe it's like a transition period as we're developing the omic systems more and learning more about those functional traits. We can move towards leveraging those technologies, but right now I think we're more on the hands-on tactical end of the spectrum, at least from my understanding or experience. Yeah. Great question. Sorry. Mic drop. Oh, thank you. Oops. I just have one, if you don't mind, 2 questions from Zoom. In your lit review or other experience, how much did you see invasive species upsetting functional biodiversity? This is Nathan. Hi, Nathan. I stayed away from most of the invasive species conversation in my lit review. And so that there is a lot out there that I just was not able to cover in this document. And in my other experience, I have not had a lot of experience with invasive species, but I'm sure there's a lot there. So that's a great question, Nathan. And I'm sorry I don't have any conclusive answers for you. And Mike, hi Mike, how can we insulate measures of functional diversity across regions or across areas with different species compositions? For example, diversity metrics would likely highlight that a southern loblolly pine plantation harbors a greater amount of functional diversity than a temperate coastal rainforest or subalpine spruce fir forest. Mike's an entomologist in forests, so this is a great question, Mike. Insulating measures, does this mean like, how do we adequately compare them to one another? I don't know if you're actively listening. Yes, okay, how can we compare them? Yes, great question. So that's where I think those indices come into play. So I highlighted the example where we had functional evenness, richness, and divergence. And I think those are becoming more commonly used indices that would allow us to do exactly what you're talking about in comparing functional diversity across different regions or areas with different species. So my answer to that would be by agreeing on some common diversity indices, functional diversity indices that we could then use in those different situations. Hopefully that answered your question. Thanks, Mike. I don't know. Yeah, a Sam question. Yeah. Sure. Right. Yeah, so the question was, did I come across anything specific to less charismatic species and local indigenous and local community perceptions of that? Something interesting and related that I did find was that a lot of times folks conducting assessments with local indigenous communities did not use the term biodiversity at all. And so I think that the way one could approach that is by finding other ways of communicating about it versus saying like, here's the Latin name of this soil bacteria we're interested in. Finding out, like, building relationships with those communities to understand what's important to them, and then connecting it to something like soil microbial diversity. I think that would be the step that the process I would take to do that. But there wasn't a lot of specific work to the perceptions. But yeah, the not using the term biodiversity, I was like, oh, that's so interesting. And I think that really speaks to developing those relationships and finding out ways to communicate with one another to achieve a shared goal. Yeah.
Thank you, Alex.
Thank you all.
The committee will meet in the room plant science 279M.
So give maybe 5 minutes to.
Yeah, a little transition.
Thank you all for attending.
Thank you for joining online.
Yeah, I was gonna try to thank you. I don't know how to use Zoom. Do I click end it?